first cells were prokaryotic cells. Prokaryotic cells are more simple than eukaryotic cells, but still must carry out all functions of living organisms.
Pilus and flagellum help the cell move, the cell wall protects the cell, and the plasma membrane regulates what goes in and out of the cell. The nucleoid region houses the DNA, and the ribosome synthesize proteins.
Prokaryotic cells are the ancestors to all organisms alive today. They have evolved over time into all the different types of cells, but they exist alone, usually as bacteria.
The cell membrane is made of phospholipids. Each phospholipid has a nonpolar hydrophobic tail and has a polar hydrophilic head. The membranes are about 5 to 10 nanometers wide, and there are proteins and cholesterol throughout the membrane. The proteins can either be integral transmembrane proteins or peripheral, which means that they're only bound to the surface. The cell membrane models the fluid mosaic model because proteins are embedded in the lipid bilayer. The hydrophilic R groups face the water and the hydrophobic R groups face the lipids. Simple diffusion is movement across a membrane from an area of high concentration to an area of low concentration. This is the concentration gradient. Small, nonpolar molecules such as oxygen gas can easily pass through the phospholipid bilayer because they're nonpolar, just like the hydrophobic tails. Molecules that are bigger and that are polar cannot be moved through simple diffusion but must use facilitated diffusion with transport proteins. Channel proteins are integral proteins that go through the membrane, and molecules can pass from high concentration to low concentration with the concentration gradient and the channel protein. Carrier proteins take a molecule such as glucose and move it from one side of the protein to the other. Osmosis is the diffusion of water, normally from an area where water is a solvent, from a high concentration of solvent to a lower concentration of solvent so that the concentrations of the solutes are balanced. Water moves in osmosis through aquaporins. Hypertonic solution, water will move to the side that has a greater concentration of the solute to balance it out. A hypotonic solution has a high concentration of water, and the water will move to the other side to make sure that it's balanced. Isotonic solutions are balanced on each side, which means that they have reached equilibrium. Active transport moves substances against concentration gradients, which moves them further away from equilibrium and decreases in entropy. This can be coupled with ATP or other energy-releasing molecules. An example of active transport is the sodium-potassium pump. It uses ATP to move sodium and potassium across the membrane against the concentration gradient. The energy from the active transport can be used in a coupled transport protein to move other molecules across the membrane.
Water potential is the likelihood of water to move from one area to another. It is commonly represented by the Greek letter psi. Water potential is calculated using the following formula. Water potential equals pressure potential plus solute potential. The way to calculate water potential is to find where the pressure potential equals 0. This means that the water potential will be equal to the solute potential. In order to find the water potential, you must first find the solute potential by using the formula -I times CRT. This formula and how to calculate the water potential is modeled in your Finding the Water Potential of a Potato lab.
Osmosis is the flow of water through a semi-permeable membrane from high water concentration to low water concentration. Osmosis explains how systems can reach equilibrium and how solutions can become isotonic. The water moves across the semi-permeable membrane in passive transport. For water to pass through the membrane, water goes through aquaporins. Aquaporins are transmembrane channel proteins that have polar passageways, allowing for the diffusion of water across the membrane.
Other ways of transport include endocytosis and exocytosis. In endocytosis, things come into the cell. Some examples are phagocytosis, receptor-mediated, and phagocytosis.
Exocytosis is when things go out of the cell in vesicles. Binary fission occurs in prokaryotic cells during cell division. Binary fission is just pinch, split, done. This is a DNA molecule shown twisted in a double helix. During DNA replication, the helix untwists. As you can see, there are two strands and they run in opposite directions as they are anti-parallel. In this case, the top strand is 5' to 3' and the bottom strand is shown 3' to 5', although we know it is read 5' to 3'. In the first step of DNA replication, helicase separates the two strands of DNA. Topoisomerase helps the untwisting of the strands by relieving strain just in front of the replication fork. At the opening, the replication fork can be shown in a Y shape. Single-stranded binders keep the two strands from rebinding to each other at this stage, and they would be somewhere around here. Then primase, an enzyme, binds to one of the DNA strands and creates a primer, synthesized from RNA bases. This section is the origin of replication, as the DNA will be made from this point on either side. The leading and lagging strands will meet here.
At the primer, DNA polymerase III binds, adding bases in the 5' to 3' direction. As shown, the leading strand is made without breaks as the DNA polymerase is moving in the 5' to 3' direction. On the other hand, the lagging strand cannot be made continuously. It must instead be synthesized in pieces called Okazaki fragments. These fragments begin at the primers, and each subsequent primer is placed further down the DNA strand. DNA polymerase III makes the Okazaki fragments. They are made in fragments because the DNA strand can only be read in the 5' to 3' direction, even though that is not the way that the polymerase moves. Once the RNA primers have been removed, DNA polymerase I inserts bases into the empty spots. After the new strands have been completed, ligase bonds the old and new strands into a double strand.